I'll introduce Jacob. Nikodym is in his final stages of the Dr. Plant Health program. Today is his final seminar. And following the seminar, we'll move into his final exam. And the seminar today, again, Jacob moved in here with us in the Dr. Plant Health program a few years ago. And he is a graduate of the University of Nebraska Agronomy Department. And he also, during his time in the Dr. Plant Health program, was in the National Needs Fellowship program as a National Needs Fellow, working on a resistance management project that we have going. So the project that he'll be talking about today, or some of the information he's talking about today, is applicable to some of the things he's been doing. So I guess with that, Jacob, you want to take it away and start the seminar.
All right. Well, thank you everyone for being here this morning. I'm really looking forward to being able to share this information with all of you. Here we go. So just a brief outline of some of the information I'm intending to cover this morning. I intend to start off with a brief history of the development of insecticide resistance, starting with the history of insecticide use, moving into some of the early days of resistance development, and then just some of the factors that have contributed to that resistance development. I then intend to talk a little bit about the current status of resistance with some of the resistance management strategies that are being employed, talking a little bit about BT crops and the role that they play in managing resistance, and then also the concern of resistance that they also pose. And then finally, just talk a little bit about some interesting challenges with managing insecticide resistance in tropical crops. And then finally, I want to talk a little bit about a few tactics that could be useful in the future for insecticide resistance management. This including area-wide management of insects, utilizing host plant tolerance, gaining a better understanding of the mechanisms by which a resistance occurs, and utilizing RNA interference as a new method of pest control. So we'll start off with just that history of resistance. So looking back historically at insecticide use, the first recorded uses of insecticidal products goes all the way back to 4,000 years ago in several ancient cultures where they used elemental sulfur dusts to protect their crops. Moving a little more recently, talking about using a variety of plant extracts, such as pyrethrum from poppies, additionally nicotine from tobacco. This was utilized widely in Europe about 200 years ago and in the Americas as colonists began to move into this area. And also in a few more recent years, we see some use of a number of inorganic compounds, including Paris green that was first utilized for control of the Colorado potato beetle as it expanded across the United States. And additionally, the use of sulfur lime and hydrogen cyanide for insect control. Moving into a few of the more modern insecticides that more people are probably aware of, the synthetic neurotoxic insecticides. We're looking at the chlorinated hydrocarbons, which were first utilized in 1939, the organophosphates, first utilized in 1944, the carbamates in 1950, the pyrethroids, which were first utilized and released in 1977, and the neonicotinoids, with the first product being released in 1991. And so with that, I'll move into some of the early history of insecticide resistance and the first reports of that occurring. So the first recorded incidence of insecticide resistance occurred in 1914, when a scientist found that an application of sulfur lime that was intended to control San Jose scale in a citrus orchard left 74% of those insects alive, even when applied at 10 times the recommended rate. And then over the following years, they found a number of other resistances to inorganic insecticides. First with the Homoptera in 1922, when other scale insects were found to be resistant to cyanide compounds, Lepidoptera in 1928, when codling moth was reported as resistant to lead arsenate. The first reported resistance in acari was in 1941, when a blue tick population was identified that was resistant to sodium arsenite. In 1942, a population of gladiolus thrips was identified that was resistant to applications of tartar-emetic. And in 1943, a population of walnut husk flies was also reported as being resistant to insecticide products. So moving on into those more synthetic, newer insecticide classes, in 1946 was our first reported resistance to a synthetic insecticide, in this case, DDT. And that was when houseflies in homes in Italy started to reemerge after those homes were treated with DDT for mosquito control. In those same areas over the next year or so, first reported in 1947, mosquitoes themselves also began to develop resistance to those insecticides. And in 1948, bed bugs in Hawaii were reported as being resistant to DDT applications. So as we can see, within the first approximately 10 years of its use, DDT resistance was beginning to emerge all over the world in a variety of insect species. Moving on to the other large insecticide classes, the first reported resistance to organophosphates was in 1947 and was found in housefly.
Talking, Janice.
The first reported case of resistance to the carbamates was in 1958 when spotted alfalfa aphid was reported as resistant to the carbamates. In 1978 was the first case of pyrethroid resistance. In this case, 2 reports with two spotted spider mite and diamondback moth both being reported resistant to pyrethroid insecticides that year. And in 1994, the silverleaf whitefly was reported resistant to imidacloprid as the first example of resistance to a neonicotinoid. So here again, we see within the first 4 to 5 years of each of these products being utilized, resistance was being reported in various insects across the world. There are a number of factors that have driven this quick development of resistance. The first of these is really the reduced use of non-chemical management tactics after the initial success of these chemical controls. So up until the late 1800s, there were a number of diverse management tactics that were utilized for insect control. These included crop rotation, residue destruction, other types of biological controls, various aspects that used those insects' biology against them to help in managing the damage that they cause. And this was largely due to the fact that there were limited chemical controls available at this time. And as a result of this, there were also high losses to pests observed in these time periods. So then in the late 1800s, initially with the success of Paris green, and then moving into the first half of the 1900s, leading up to the boom of synthetic insecticides, we saw a lot of increased interest in those chemical control tactics, often at the expense of focus on those other non-chemical controls. People started utilizing chemical controls rather than relying on pest biology to manage those pests. And in the same time period, we saw a lot of increased interest in utilizing plant resistance in control as well. And in the short term, this was a very effective and easy solution to the insecticide pest problem, and initially seemed like it would be the best way to control insects going forward. Another factor that has driven the evolution of resistance is the use of repeated, often prophylactic applications of insecticides. This leads to the presence of very persistent insecticide residues in the fields and has the benefit of preventing establishment of insecticides and inherently preventing damage to crops. However, this also leads to a continuous selection pressure being applied on those insects to that insecticide mode of action, generation after generation, which leads to rapid buildup of resistance. An additional factor of this same situation is the danger of sublethal exposures. As these residues degraded, they became less and less toxic to those insects, therefore allowing more insects to survive and any only partially resistant individuals to survive and pass on those resistance alleles. In addition, this led to problems with hormesis, where a small dose of a toxin actually promotes growth of an insect rather than impeding it. So we saw this rapid accumulation of insecticide resistance alleles in these populations. Just as an example of how this process could play out, we see in the first generation prior to application, there's mostly susceptible insects on that leaf. We see maybe a little bit of already existing resistance due to standing variation in that population, but the majority of the population is susceptible to application. And therefore, after that application, very few of those susceptible insects survive, but we do see that that resistant individual has survived. So then that leads to an increased prevalence of those resistance alleles in the remaining population. So after a few cycles of this, several generations later, we see here in the later generation that a much larger proportion of that population is resistant to that insecticide. And after application, the vast majority of the insects that were present before the application remain present, and we see a loss of control by that insecticide. And another final factor that I believe has driven resistance over the years is a loss of natural enemies as a result of insecticide control tactics. This leads to resurgence of primary pests as those populations are able to recover more quickly than the natural enemy populations. And in addition, it releases secondary pests from natural enemy control and also from the competition that they may have been experiencing with those primary pests. This allows them to become a much larger concern as a pest resistance issue, and also causes them to have that exposure to that insecticide and allows for resistance development over the generations. So all of these factors combine to contribute to what has been referred to as pesticide treadmill. We start out with a chemical that's effective and is being utilized well for control. After a few generations of this type of repeated application of this insecticide, we start to see a loss of efficacy in this insecticide. With this lost efficacy, we try to accommodate for that and maybe increase the rate at which we apply that insecticide, which works for a while until, again, reduced performance starts to occur. We're losing control of that insect with even that increased application. Well, by now, it's probably been long enough that a new chemical has been brought to market that's effective against this pest. So we're saved. We've got a new product that will allow us to control those insecticides, which just brings us back into that same cycle. And we just continue moving on the treadmill, moving through new chemistries as they're made available until they're no longer effective. And this problem was observed beginning in probably the late 1950s into the 1960s. Many scientists began to see this issue and call for changes in management tactics. And we're calling for more integrated pest management techniques. And in the 1970s and 1980s, the USDA began to sponsor a number of integrated pest management programs. And these programs have been associated with a 70 to 80% reduction in the use rates of some insecticidal products. This is possibly due to the replacement of some of these products with simply other products, but there was some reduction in the overall usage of pesticides. And another, just a great success story of the integrated pest management system can be found in the control of codling moth in the California pear industry. In the 1950s through the 1970s, chemical control was the primary method of control for this pest. And toward the end of the 1970s, they were starting to see increasing damage and increasing pest pressure as those insecticides became less effective due to the development of resistance. As a result, they turned to an increased focus on utilizing integrated pest management, which led to up to a 96% reduction in organophosphate use in some situations, while at the same time helping to increase pest management efficacy and reduce the damage that they were seeing to their crops. Moving on from there, we'll move into some of the current status of insecticide resistance. And just to put this problem in perspective, this graph is based on data from 2015 that shows the 12 most resistant arthropods at that time. As you can see, two-spotted spider mite, diamondback moth, and green peach aphid are all very serious resistance concerns, having greater than 70 modes of action that they have developed resistance to, greater than 70 active ingredients that they've developed resistance to. And putting in perspective of those three, some of these others don't seem quite so bad, like the Mediterranean climbing cutworm at the bottom there, with only 38 chemicals that it's developed resistance to. But thinking about it, even 38 chemistries, that's a lot of products that are not available at this point for control of that species. And just to look at how this trend has occurred over the years, we see here this graph starts from the beginning when that first case of resistance was reported in 1914. Over that first 30 or so years, there really wasn't a lot of resistance developing, just a few cases here and there. But into the 1960s through the 1990s, there's a very rapid increase in those numbers of species. In 2015, there were over 500 species that had developed resistance to approximately 325 insecticides and 5 insecticidal plant traits. Whereas in 2021, those numbers have increased to over 600 species, 300 to 349 different mode of action, insect chemistries, and 13 different insecticidal traits. And in response to this rapid development of resistance, a number of groups have been formed to help inform the growers and those managing these pests of the ways to manage this resistance concern. One of these groups is the Insecticide Resistance Action Committee. This group was formed in 1984 and is comprised of members from all of the major insecticidal chemistry manufacturers. Their goal is to promote the understanding and management of insecticide resistance, and they are considered one of the foremost authorities on insecticide resistance management and on insecticide resistance. They also are the producers of the most widely utilized mode of action classification to inform farmers of proper rotations and what products they're actually using. So insecticide resistance management, there are a number of factors that go into properly utilizing these plants. The first aspect of this, and probably the most important, is properly planning the insect control tactics that you're going to use in order to manage this damage to your plants. If you're with proper planning, you can avoid a number of potential issues with over applications, incorrect applications, things like that. Another important aspect of resistance management is utilizing integrated management tactics. By bringing some of those cultural control methods, biological control methods back into the management process, we can help to reduce selection pressure on those insecticidal chemistries and extend the life for which they are effective. Another important part of this is rotation of modes of action, whether that be seasonal or within the season. And within the season is especially important when an insect undergoes multiple generations in a year. As you can see in this graphic, there are a number of different ways that modes of action can be rotated through the years and through the generations. And what are considered to be the most effective rotations are when the same mode of action is used for every application to a certain generation, and then rotated to a different mode of action for the next generation, or when a different set of modes of action is used for each generation in order to prevent a single generation from being exposed to multiple modes of action and then having that same multiple modes of action exposed to the next generation, which can offset the benefits of that rotation. Another option that has been considered is mode of action mixtures, but these are generally not recommended for management of resistance. This is due to a lot of the same issues that would be for rotating within a generation rather than in between generations. We would simply impose selection pressure for multiple modes of action on each subsequent generation, which leads to the danger of double resistance developing, in which the insect is able to be resistant to both modes of action being utilized. There are a number of factors that impact the management of mixtures when being used in pest control. Some of these considerations include considering the individual effectiveness of each of the products in that mixture to control that insect, making sure that the individual modes of action of those products are indeed different and we aren't just applying two different products with the same mode of action. We also need to consider the danger of cross-resistance developing between those different products in order to make sure that there isn't we aren't just selecting for a single resistance that allows for that insect to be resistant to both. And finally, we need to make sure that if we're utilizing mixtures, both of the products in that mixture have similar residual periods. So there's no point during the degradation of that product that only one of the two products is still present in the field, encouraging selection for just that one mode of action. Moving on into BT crops, these have been a very effective tool in the integrated pest management toolbox in helping to manage insecticide resistance. These crops were first developed and released in the mid to late 1990s. The first product release was new leaf potatoes by Monsanto, which was targeted to control Colorado potato beetle. In the late 1990s and the years shortly following this, we saw there were releases of corn to control European corn borer, and cotton to control cotton bollworm and tobacco budworm. In 2002, the first insecticidal product to contain pyramided traits, which are multiple traits to control a single insect, was released, with cotton expressing Cry1AC and Cry2AB, both targeted for control of cotton bollworm. Then the next year, in 2003, the first crop with stacked traits, or traits that are targeted at different pests, was released, with being corn with Cry1AB1 to target the European corn borer, and Cry3BB1 to target northern and western corn rootworm. Over the period of 1996 to 2006, the use of BT crops, particularly cotton and corn, was associated with a reduction of 101.5 million kilograms of insecticide active ingredient, which was a great help to helping to manage that growing resistance problem as we remove those insecticides from that selection process. But just as with insecticides, BT also provides its own resistance concerns. And as a result, these products were released with resistance management plans. And it is important to consider that these resistance management plans were designed with a specific pest in mind. So in many cases, if other pests were also present in that field and being impacted by the BT, they may not have been considered as heavily in developing that resistance management plan, and it may not have been as effective in managing resistance to those species. This is also seen with the release of corn rootworm targeting corn plants, which still utilize the same resistance management plans as European corn borer plants, which caused some problems down the road. The main strategy that was utilized in these resistance management plans was the high-dose refuge strategy, in which the plants expressed the BT proteins at a high enough rate to kill 99% of susceptible heterozygotes. This ensured that any heterozygotes resulting from the mating of a resistant and a susceptible individual would be killed by a following dose of that BT. This was important due to the combination of the high dose with the use of a refuge that allowed susceptible individuals to survive and mate with those resistant individuals that may come out of the traded plots. There are a number of different structures of refuges. A structured refuge typically was assigned to be about 20% of the size of the traded field, while mixture refuges, in which untraded seed is mixed in the bag with traded seed, usually require approximately a 5 to 10% mix of refuge seed with the traded seed. And one primary concern with BT resistance in the Midwest and the Corn Belt has been resistance by corn rootworm to BT. This is due to a number of factors. One of these is a lack of refuge compliance, in addition to that problem of the refuge management plans being designed to work with European corn borer rather than corn rootworm. But this lack of refuge compliance was due to either the inability or unwillingness of producers to comply with these requirements. For example, in a 2001 survey, while 90% of producers indicated that they had been following proper refuge procedures, only 71% of them properly defined the actual refuge requirements for their crops. In addition, with corn rootworm in particular, there was a failure for these hybrids to achieve a high dose of the insecticidal BT proteins. These corn rootworm products were identified primarily as being a low to moderate dose, which did not sufficiently kill those heterozygous susceptible individuals, allowing them to survive and pass on their resistance alleles to following generations. Looking specifically at the benefits of refuge, there is a very good case study within corn rootworm management that indicates how successful refuges can be. In corn rootworm, there were a number of organochlorine insecticides that developed resistance here in Nebraska. This was reported in 1959 and was largely a result of these insecticides being applied to the soil in broadcast applications covering the entire soil surface and having very long residuals within the soil, leading to complete and continuous selection pressure against all of the larvae in those soils. By contrast, resistance has not been observed to other soil insecticides that have been used for corn rootworm management. This is likely due to the fact that these products have been applied as in-furrow or banded treatments, which allows those corn rootworms that exist outside of the treated area to feed on corn roots that grow outside of that area, acting as a refuge between the rows and allowing the maintenance of susceptible alleles in those populations. Moving on from BT to the management of tropical agriculture pests, this is a serious concern due to the fact that continuous production systems in these areas can lead to pest management practices on the individual field level to be less than effective, especially crop rotation, as when one field is harvested, it's likely that another field down the road has already been planted to a new crop that could also be a host for that pest species. As a result, it has been very necessary to develop IPM strategies for proper resistance management in these areas. This includes the development of proper economic injury levels to inform when insecticides are actually needed for management. It is very important in these areas to implement proper use of BT refuges when BT crops are utilized, and also using burn down treatments to control any alternate or volunteer hosts in the area in order to ensure that that pest cycle can be broken up as much as possible. So having discussed that current status of where resistance is at, I'd like to move forward into what might be some future tactics for the management of insecticide resistance. just as a quick aside, looking forward, this is something that needs to be considered because the cost of developing insecticide products is very much increasing. In 1960, the estimated cost of bringing a new insecticide product to market was about $4 million, which when adjusted to 2012 inflation numbers, equates to about $31 million. By contrast, in 2012, the estimated costs of developing a new product was over $250 million. It is not sustainable to continue utilizing insecticides the way we have and allowing resistance to develop the way that it has in the past. Insecticide resistance management is a necessary part of maintaining the sustainability of insect control in agriculture. One area where I think will be very beneficial to continue studying in order to manage insecticide resistance in the future is increased looks at the use of area-wide pest management to reduce the insect pressure in growing areas. This is especially potentially useful in those continuous cropping areas that I discussed a little earlier. This has seen some success in southern Queensland and Australia in the late 1990s. They saw a lot of problems in brassica crops with diamondback moth. And as a result, it was recommended to producers that they utilize what was referred to as a brassica break, in which for approximately a 3-month period, they not grow brassicas in these areas in order to break up the life cycle of the diamondback moth. And with fairly high levels of adoption, this saw a lot of success, and the pest pressure in the areas was reduced. And they, in some cases, actually with testing, saw that some of the pesticides being utilized there actually had reduced LD50s after these breaks. After a number of years of these breaks, as some of the resistance in those populations was seen to be abating. Even though this was a successful process, after a few years, those farmers stopped utilizing it as much. They went down to maybe a 1-month break, and after a while stopped utilizing these breaks entirely. And this is an example of why, in many cases, in order to facilitate the long-term use and efficacy of these types of area-wide pest management programs, it will most likely be necessary to integrate these processes into policy choices, and aiding in ensuring that there is widespread compliance with these regulations, which is necessary for them to be effective. Another situation where area-wide pest management has been effective in managing insect pests is in the use of and control of the codling moth in the Western United States and Canada. These programs have helped to reduce pest pressure and reduce insecticide usage in a number of stone fruit species. One aspect of area-wide pest management that is very useful and has played an important role in those codling moth programs has been the use of the sterile insect technique, in which sterile male insects are raised and released en masse into these areas and are allowed to be competitive reproductively with the native populations. As a result, this reduces fecundity and population sizes for the following years and allows for reduced pest pressure and, in some cases, even eradication of insects. This has been seen with the New World screwworm through the 1990s and continuing to today. The New World screwworm has been eradicated from North America down through Mexico and is still being maintained as eradicated in those ways through releases of sterile males in Panama. Another more recent situation has been the eradication of pink bollworm from the southern United States, utilizing the sterile insect technique. While this is a very powerful technique for the management of pest insects, it does have its limitations. The primary one of these is that it requires a very high economic input. It is not a cheap process to mass rear and sterilize male insects in such a way that they remain reproductively competitive within the natural populations. And another limitation of the sterile insect technique is that it is very dependent on the mating tactics of the insect that's being targeted. The sterile insect technique is only really viable if female insects of that population only mate once, or at least if they mate multiple times, that they hold on to that sperm and utilize it randomly for fertilizing eggs. This is because in these situations, if that female mates with a sterile male, she either will not produce any viable eggs, or she will produce a reduced number of viable eggs due to those eggs that have been fertilized with a sperm from a sterile male. On the contrary, if this is a species where females mate multiple times with multiple males, this is less effective due to the fact that even if a female mates with one of these sterile males, she may then again mate with a fertile male and is able to produce offspring just as she normally would. Another aspect of, and a tactic that was once more widely understood and over the years has not been as well studied as many other insect management techniques, is the use of tolerance for insect management, host plants tolerance. This fits into an important aspect of IPM in which the focus is placed on managing the stress of the host rather than controlling the pest. This means that rather than trying to completely eliminate the pest from the area and from causing any damage, we simply keep the pests at a low enough level that they are not able to cause an economic level of injury to that host. And tolerance helps in this process by allowing plants to better recover from damage that they may sustain. A couple of examples that have been observed with this, in certain circumstances, raspberry has been shown to have increased growth after experiencing feeding from aphids, which allowed those raspberry plants to overcome the damage that that aphid feeding caused and maintain their growth through that stress. In another situation, a sugarcane has been shown to have increased growth of tops, stubble, and roots in response to white grub feeding on the roots, therefore overcoming some of that damage to the roots and allowing the plant to continue to yield successfully. As I stated, proper use of insect tolerance in host plants allows a reduced use of other management tactics, therefore reducing the selection pressure imposed by those tactics on those insects. And in addition, tolerance poses a very minimal risk of resistance developing due to the fact that it doesn't really select those insects for benefits, because in these situations of plant tolerance, the insects are able to complete their life cycle and feed just as they would in any other situation. But the plant is simply able to overcome this. As a result, selection pressure is not applied and resistance is not likely to develop. Another big aspect of proper management of insecticide resistance moving forward will be further understanding the mechanisms by which resistance develops. There are a number of primary mechanisms through which resistance occurs. This includes increases in metabolic detoxification of the insecticidal products using a number of enzymatic processes in the insects before they reach their target, before the chemical reaches its target site. Genetic alteration of the target site itself in order to prevent the binding and action of that insecticidal product, and reduced penetration of those insecticidal products through the insect cuticle in order to prevent that product from reaching its target site. These mechanisms of resistance can be studied in a number of ways, and the mechanism through which resistance occurs has a large impact on the best way to manage that resistance. In addition, understanding these resistance mechanisms can help us to understand what dangers of cross-resistance may exist in these populations. This is especially true when we consider resistance that occurs through detoxification of chemicals. When this type of resistance occurs, many of these chemicals can be detoxified by the same enzymes in that insect's metabolic system. So as a result, if resistance has developed to one mode of action through detoxification, then this can contribute to resistance to a different mode of action that is completely unrelated to that initial mode of action and that the insect has never been exposed to, presenting a new problem of resistance in which resistance may occur when it's unexpected. There are a number of ways to study the mechanisms by which insecticide resistance occurs, but a couple of the more recently developed ones include RNA interference, in which we utilize a double-stranded RNA molecule that is injected into or ingested by an arthropod that targets and breaks down mRNA genes that are homologous to the sequence of that double-stranded RNA. This prevents the expression of that gene. And if this is used to target metabolic enzymes, then we can, after utilizing this treatment, expose that insect to a pesticide that it may have been resistant to previously and examine what the impact on that resistance has been, therefore helping to determine if the gene that was knocked out had any impact on the development and existence of that resistance. Another option is utilizing the CRISPR-Cas system, which utilizes a guide RNA sequence to target a specific portion of the insect's DNA. This allows for creation of double-strand breaks in that DNA strand, which in turn are repaired by the cell machinery. And through this repair process, new mutations can be artificially induced in that gene, which can either knock out a certain gene or introduce a new gene that was previously not present in that space. As a result, we can artificially induce a mutation in that insect and then utilize that artificially induced mutation to confirm or refute whether that mutation is necessary for insecticide resistance, therefore helping to define what are the causes of resistance in a certain species. In addition to being able to be utilized for management of insects, RNAi has, or in addition to being used for studying the causes of resistance, RNAi also has potential to be used in management of insects in the field. As it is a naturally occurring gene regulation process in many species, it is a potential target for management techniques. If we design a double-stranded RNA molecule that targets a gene important for insect survival, we can then silence that gene that is necessary for survival and cause mortality or other impacts within that insect. It is important to consider, however, that resistance, just as with all of the other products we've talked about, could still be a concern. And in fact, resistance that was initially selected from a field population in Illinois in 2011 and then selected further in laboratory experiments has been shown to the corn rootworm targeting double-stranded RNA molecule that targets DB-SNF7. This resistance was identified to occur through reduced uptake of the double-stranded RNA molecules, and as a result, it is not specific to this double-stranded RNA molecule, which indicates that it is likely that in order to properly manage potential resistance to RNAi, we will need to utilize and rotate this product with other management tactics, utilizing those integrated management techniques in order to prevent and slow the development of resistance. There have been a number of RNAi targets that have been studied and are being considered for utilization in insect control. First, for the corn rootworm, as I mentioned, DV-SNF7. This is the most studied thus far target for RNAi and has been commercialized as SmartStax Pro corn rootworm by Monsanto and now Bayer, and should be available within the next few years for producers. Another couple of corn rootworm genes that have been studied a little further is DVBOL and DVVGR. These two genes both target these two double-stranded RNA constructs both target reproductive genes in the corn rootworm. And as a result, when they are silenced, they cause reduced fecundity, reducing the population size of the following generation. Another gene that has been explored is SEC23, which is a gene that has an impact in the formation of cellular structures in corn rootworms. And as a result, if this gene is silenced through RNAi treatment, it leads to mortality in those insects. Another species that has been fairly extensively studied for control with RNAi is Colorado potato beetle. There have been 27 potential target genes that have been identified for this species, including ECR, which is a molting gene, and as a result causes mortality in larvae when it is silenced, though it has minimal impact on adults due to the fact that they have finished molting by the time they become adults. Another potential use for RNAi that has been studied in Colorado potato beetle is to utilize it to improve the efficacy of other management products. For example, the gene CNCC regulates the expression of a number of detoxification genes in the Colorado potato beetle. As a result, when CNCC is silenced using RNAi, those beetles are more susceptible to treatments with imidacloprid. Another example of this is prohibitin, which regulates transcription of a number of genes and has a role in mitochondrial function in Colorado potato beetle. And when it is silenced, it actually leads to an increase in the efficacy of the Cry3AA BT protein against those individuals. There are a number of other drawbacks to the use of RNAi for insect pest management. One of these is the fact that oral delivery is the most practical way to utilize these for pest control. So as a result, the most effective way to introduce these products to the insects will be through genetically modified crops, because broadcast spraying will likely be less effective due to the inability to utilize contact applications like we see in a lot of insecticidal products. Another potential drawback is, particularly for oral delivery, RNAi is highly efficacious against coleopteran pests, but research has shown that it is not as effective against pests in the orders Lepidoptera and Hemiptera. This is due largely to a difference in the way that RNAi is taken up from the double-stranded RNA molecules are taken up by these insects, and the way in which RNAi responses move systemically through these insect species. And finally, RNAi has a very short persistence in the environment. As a result, there will need to be research to improve this persistence in order to ensure that these products can be used effectively and have some form of lasting control in the environment. And there isn't a danger of these double-stranded RNA molecules degrading before they are able to have any impact on insects, particularly in situations where a broadcast spraying application may be utilized to deliver them. So just moving on to a few quick conclusions. I don't think I need to tell most people here that Insecticide resistance is a very serious concern facing agriculture today. It is a problem that will need to be addressed in order to maintain the sustainability of insect management in agriculture. And an important part of that insecticide resistance management will be the use of IPM programs. Utilizing integrated control helps to reduce selection pressure for insecticidal products and sustain the effective life of those products. And I do believe that a combination of utilizing new technologies as they develop and looking back to the past at some of the classic strategies that have been effective for insect management will be a very important part of being able to sustainably manage insects in the future. Thank you, and with that, I will take any questions that anyone may have.
If there are questions, you can either type them in the chat or you should be able to unmute yourself and ask it directly. Any questions out there? There's one. There's one.
So Nathan's asking, after resistance is established and chemistry moves out of use due to that resistance, are there cases of susceptibility returning? And yes, this is very possible. In some cases, there is a fitness cost associated with that insecticide resistance, meaning that that resistance causes those particular insects to be less competitive in that population in the absence of that stress. So in those cases, it is very possible that we can see that susceptibility beginning to return through the process of those insecticides not being utilized. I didn't personally research a lot into that susceptibility, any specific cases of that susceptibility returning, but it is definitely a very possible situation that can occur.
Any other questions? Hearing none, I guess we can call that good then. We want to thank Jacob for his presentation and thank you for attending. Okay. So thank you very much. And we all acknowledge, I guess, Jacob's efforts here. Thank you.